Good evening, everyone, and welcome to the first Atom Talk of 2021. Thank you all for joining us. I hope that you had a peaceful and restful festive period and you're raring to go. It's a brand new year. The days are getting longer. The vaccines are rolling out. We're full of hope. So let's have a good evening tonight. Before we move on to the main event, I've got a few admin things to get through. We will also be having science snippets this evening, so if you do have any ready and raring to go, please pop them in the chat now so that I can bring them up when I get to them. And I know that Alphonse, our committee member, has a few to bring up himself as well. So just really quickly then, the first thing I wanted to bring up was I've been asked by the committee of the Atom Festival to pass on a message. Obviously, ordinarily at this time, we would be starting to advertise the festival, get you to get your tickets and get excited about it. This year, it will not be happening in March. I've been informed by the festival committee to please inform you that this year, they won't be hosting the festival in March. They do intend to host the festival, however. It will be moving to June or July. I forget which one of July. Alphonse is nodding at me. Maybe sometime around then. It will be in the summer. And I will send this in an email later to the mailing list, so you will have accurate information, not the mad ramblings of someone on a Thursday night. But it will be in the summer this year. At the moment, they're not entirely sure what format it will be in. It's likely to be shorter than it usually is. They are planning to have things online. Obviously, if they can do things in person, they will, but only if it's safe to do so, and more information will follow. So now that I've told you that, the other thing I just wanted to bring up was obviously in my email, I requested some assistance for the committee. Our numbers appear to be dwindling, and especially being online, it does take a lot more effort actually for the committee to keep this going. I have had someone come forward, and that's great. I'm going to be speaking to him soon. But if anyone else feels that they will be able to spare a very realistically quite a small amount of time to help out in any way with the committee, either with publicity, with some of the roles of secretary, or literally anything that you think you could help out with, please do get in touch. We only have a meeting once every 2 months, and the meeting itself is normally only about an hour and a half, so it's not a huge amount of time to commit. And as I said in my email, once we start seeing each other again, you do get to have dinner with the speakers before each month's event, which is quite fun, and it's nice to chat to them normally. So just having a look for the science snippets. Jutta has informed me that the dates for the festival are the 11th and 12th of June. Thank you very much, Jutta, for that. Okay, so if you've got any science snippets, please do pop them in the chat and I'll bring them up. I will just bring Alphonse in. Good evening, Alphonse.
Hi, Daniel. How are you?
I'm good, thank you.
Yes. I found a few ones. So if you can put my screen in the show. Yep. Yes, there you can actually see it. So there was a study which was recently published that there's actually quite a bit of drinkable water, so fresh water, which is hidden under the oceans. So people in the past have found that by drilling for oil just by accidents, and that may actually help solving in some of the regions the problem for people to get fresh water, either for watering their plants or actually for drinking. And you can see like the big blobs there, that are the areas where the studies have found this water to be. And some of them are actually close to the brown regions, which is where there is not much water or not much rain falling at all. So that can help. It's not quite clear how easy it is to get at that water, but I thought that's an interesting study, which in the future may help us to grow more food.
Yeah, just looking at the Australia blue blobs, I think they would be very useful if they could be exploited.
Yeah, and also some things in South Africa.
Yeah,
It's surprising to see Greenland so brown? Because there's plenty of packed ice, but apparently no rain or snowfall ever.
Yeah.
Yeah, that was one. And I have another one which is entirely different. Virgin Galactic. So what you can see here is actually a little, or maybe not so little rocket, because what you see here is a Boeing 747-400. And this jet was cruising at 11 kilometers height, and at that point then launched this rocket, which you can there see mounted under the wing, and brought a satellite into orbit at 500 kilometers. And that's the first ever satellite that has been launched kind of horizontally and brought into Earth orbit. So that's also quite an achievement. That's still a little bit behind other endeavors of private companies, but I think that's a good step forward.
Definitely. Okay. Don't think we've got any more coming through on the chat. Did you say you had a third one?
Yeah, I have another one. Let me see whether I can make this a little bit bigger. So do you see what this is?
Looks like some woodland.
Yeah, and it's a satellite image. It's a high-resolution satellite image, not taken by the satellite we just talked about, but some other, probably what previously would have been a spy satellite. And they're now using that and image processing to find elephants. So all these yellow squares are elephants. So now you can count these animals from space, thousands of square kilometers a day, and get a good count of animals. And they use special kind of artificial intelligence to actually identify those in the pictures.
Nice. Very cool.
Yeah, I thought so.
Yeah. Okay. It doesn't look like we have any more snippets coming through on the chat this evening. Thank you very much, Alphonse. So if anyone does have any more, then pop them in and I can obviously bring them up later in the evening. But now I would like to introduce our speaker for the evening, Dr. Doultsinos. Dimitrios, originating in Athens, he received clinical training at the University of Sheffield before reading biomedical research at the University of Birmingham. He then went on to get his Master of Research from Imperial College London in clinical research and translational medicine. His research segment of his Master's was focused on ALS, but it was secondary to an MHRA qualification in clinical trial design and management. Following that, he went on to gain a Marie Curie Fellowship at INSERM in France, where he also got his PhD in oncology, where he was looking at targeting the unfolded protein response in brain cancer. Dimitrios is now a research fellow at the Nuffield Department of Surgical Sciences, and he's here to talk to us today about how protein quality control governs health and disease. So, okay.
Good evening from the Churchill Hospital. Thank you very much for the introduction and for having me to give this talk. So we'll be speaking about proteins and how protein quality control governs health and disease, and about how protein production and protein structure can lead to discovering candidates and targets in different diseases, and how we can approach those to make patient life better, and how clinical trials are designed for such potential therapeutics. So I thought about starting with the drug discovery timeline. There is a significant chunk of time at the very beginning of the drug discovery timeline that lasts for about 6 years, where medics and clinical researchers try to figure out, based on a clinical problem, whether there are any biological targets that can be exploited. So It usually starts with patients coming into the clinic. We've got a particular disease. Then data from those patients are analyzed in order to figure out which are the most important biologies that are involved in that disease. And then through those, dissecting them even further, we find biochemical pathways that are of paramount importance in the disease, and then reverse engineer some targets and some potential drugs. And then these are tested on either biological models, like cell models, or on animal models or humanized models. And then if these show promise, then the next step is to apply for a clinical trial, which lasts for less than a year. And if the application is successful, then the actual clinical trial starts, which has multiple phases ranging from 1 to 3, where one is mainly toxicology, so we're trying to figure out whether the drug is toxic or not, and then moving through the phases into larger and larger cohorts of people to figure out if it has a beneficial effect. Then if the trials meet their primary outcomes, the primary objectives, then there is governmental review in every country that we're seeking approval, and then manufacturing can start. And in fact, the whole process, phase 4, doesn't actually finish until the drug is no longer on the market, because there is a lot of post-market surveillance and reporting of adverse events throughout the lifetime of the drug. Now, in the very first part of the talk, we'll focus on that first 3 to 6 years and basic research and translational research, and how we can get from a target to a drug. So why proteins? Proteins are quite important in our everyday lives. We read, we write, we act, we have meals, we have to take medicines, fight off infections like the cold and the flu, exercise, among a myriad other functions that the human body actually carries out. And in order to carry those out, the body needs a certain degree of structure, interpretation of information, communication between the different organs and different cells, and a certain degree of execution, how to actually function. And the molecules that govern all these processes and carry them out are proteins, and they include enzymes, hormones, receptors, etc. These are all types of proteins. So the way that a protein functions, its very specialized and important function, is inextricably linked to its structure. The structure always starts as something incredibly simple. So a chain of amino acids, these are the building blocks of proteins, and they form let's say a very simple linear squiggle. So that's a polypeptide chain. Now that starts contorting into specialized shapes like alpha helices or beta pleated sheets, and they form a secondary structure which folds even more to produce the tertiary structure, and then a quaternary structure is the final product. Now every single protein has a particular structure, and that's very important for its function. And it's quite important for the rest of the talk to keep in mind that certain types of proteins, like enzymes, have little pockets. So those pockets in the structure allow substrates, small molecules, to bind onto them and then help them carry out that function. So without those, the protein or the enzyme itself cannot actually carry out its function. So how are proteins made? Proteins are made within every single cell in the body, and the cell itself and protein production is not dissimilar to a factory that makes any given product. So you will have a blueprint for all the products that the factory is making, and then from that blueprint, there will be individual manuals extracted for every single specific product. Then that manual is read, processed, and understood. And then that gives rise to the assembly line of the product, starting from a very simple material to more complex materials to the final product. So in this case, you'd have a little bit of metal, which would be made into metal sheets, which would eventually form a metal ladder. Now, then that at the end is packaged and exported, and the whole process is actually supported by energy, by a generator. And a cell is almost exactly the same. The blueprint for protein production resides within the DNA, in the nucleus. And then mRNA is the manual for each individual protein. So it resides from the DNA, it goes into mRNA as a single manual for every single protein, and that is processed and interpreted in an organelle called a ribosome. And from that, we get that simple chain that we spoke about before, that chain of amino acids, the simple polypeptide. And that's the start of that assembly line, which happens on the factory floor, which in the cell's case is a very important organelle called the endoplasmic reticulum. So in the endoplasmic reticulum, those proteins start contorting, they start forming more and more complex structures until they are complete. And then in another organelle called the Golgi apparatus are packaged and exported or used internally, because as you would expect with any given factory, some of the products that are made would be useful for within the factory itself. And like a proper factory, there is an energy supply provided by energy-giving organelles that are called mitochondria. So protein production is an assembly line. Proteins are very diverse and are essential to life. They have very complex structures, and their structural blueprint resides within the DNA. Protein production is an assembly line of many different steps, and there are quality control safeguards for each step. And it's important to remember that each cell has a maximum production capacity. So how does a factory work? As usual, there is supply and demand. So for example, if we have a meal, then we're demanding from our pancreatic cells to make insulin, which is a protein. So when that happens, there has to be an upscale of insulin production in order to meet the demand. Likewise, if we get an infection like the flu or a cold, the immune system has to wake up, it has to upregulate its production of receptors in order to recognize the pathogen and mobilize different cells in order to fight it off. That also needs an upregulation of supply and demand of protein production. And as always, you need to have quality control, because like any production line, there will be some correctly produced products and some not. So for example, if we would need our cells to make spanners, we want them to make them correctly rather than either misfolded or completely incorrectly. And then if that happens, there needs to be a troubleshooting mechanism in order to fix that. So if we get any misfolded or incorrectly folded proteins, there needs to be a mechanism to fix that problem. So how can these misfolded and incorrectly folded products or proteins or spanners arise? So we can either have problems in the blueprint. So these are what are called mutations. So if there is a problem with a manual, if you have a faulty manual, then you're going to have a faulty product. And also there are some mutations that also upscale production. There are gain of function mutations. So they would lead to an upscale of production as well as giving faulty products. And then the second one is simply increased demand, because the more increased production you have, the more increased chance you have of something not coming out quite as right. So in the end, what you get from both of these processes is an overaccumulation of misfolded proteins in the endoplasmic reticulum. That important organelle we spoke about before, which is the factory floor, is where the assembly line is taking place. And this leads to something called endoplasmic reticulum stress. So the cells start getting very stressed about the whole situation. So what can the cell do about it? Because suddenly there's a bunch of wrongly folded spanners everywhere on the production floor. So firstly, the cells need to detect stress. And don't worry about the names of these boxes. They're not particularly important, but what these sensors do is sense the accumulation of misfolded proteins on the endoplasmic reticulum, as well as testing the bonds between the structures of the proteins themselves. So if they're on the right place, if they're at the correct conformation, and so on and so forth. And then following that, we have an attempt at stress resolution. So these three sensors signal to a certain group of transducers. Now, again, the names are not particularly important, but PERK, RE1, and ATF6 form a process that attempts to resolve that stress called the unfolded protein response. And what that essentially does is ring HR that have the and head office that have all the blueprints and say, we need to increase production capacity, we need to increase quality control, metabolism, secretion, folding capacity, increase the immune response, because the factory floor cannot cope. So we need to overcome those problems. So what can the outcomes be? Because endoplasmic reticulum stress can happen on in every single cell all across the body. So in the lungs, in the brain, in the heart, depending on the function, ER stress can occur. And in each case, the resolution attempt happens through the unfolded protein response. And there are three, essentially, there are three outcomes of this. Most commonly, everything is resolved, life goes back to normal. So the stress is resolved, the misfolded items are either refolded or binned. Now, there are another two outcomes that may be problematic. One of them is that the mechanism is actually overwhelmed. There are so many misfolded items in the endoplasmic reticulum that the system cannot cope, which means that then it programs itself for cell death, so it self-destructs. That process is called apoptosis. And then there is another potential outcome where we get an over-adaptation to this stressful environment. And then this adaptation-led increased stress capacity sustains normally unsustainable levels of protein production, and usually faulty protein production as well. Now, those last two potential outcomes can give rise or support really serious diseases. First one is cancer, including brain and prostate, and it's very common across the globe. I have dealt with prostate and brain cancer throughout my career, and there are some high-profile cases like U.S. politicians, including John McCain and the son of the current U.S. president, Bob Biden, and as well as Bill Turnbull, who has the Classic FM show that, so the former two had brain cancer and he has prostate cancer. And brain cancer, for example, like glioblastoma multiforme, is a very fatal tumor. The 5-year survival is dismal. Only less than 3% survive for more than 5 years after diagnosis, and it's a highly invasive tumor. So basically, it makes it very difficult to completely cut out, which means that we always leave little bits of tumor behind in the brain, which makes it highly recurrent. So it means it comes back all the time. And certain biochemical mechanisms within it make it resistant to therapy, which includes chemotherapy and radiotherapy. And then on the other side of the spectrum, we've got other UPR-driven, unfolded protein response-driven diseases, like amyotrophic lateral sclerosis, which is a muscle wasting disorder. So it affects motor neurons, which originate in the motor cortex in the brain and tell your muscles to move. So it's very debilitating, and usually patients die from respiratory failure because the muscles that govern our breathing, they stop working because the nerves that make them move stop working, they die. And again, there were quite a few recent news articles about people who were diagnosed recently with ALS, including Doddy Weir and Rod Burrow. And of course, Stephen Hawking lived with the disease for many years, and it was quite debilitating. So what is the role of the UPR in these diseases? Now, in cancer cells, we have, because cancer cells are very metabolically active, they need energy and protein all the time. So the demand is massively increased for folding proteins. And also, cancer cells acquire mutations all the time. So both of these drive that accumulation of misfolded proteins in the cell. Now, motor neurons also acquire different mutations that correspond to an overaccumulation of misfolded proteins in their endoplasmic reticulum. And in both cases, we've got the unfolded protein response trying to resolve the matter. However, in the case of cancer, we've got adaptation, whilst in the case of motor neurons, they can't cope, which means that in the case of cancer, we get uncontrolled growth, whilst in the case of ALS, we get motor neuron death, which means that we've got two very contrasting conditions with potentially the same regulatory underlying biology. So it's quite clear that we have a target. We would be able to target the unfolded protein response, and protein production quality control is supervised by the UPR. It is a very finely balanced process, and if this process is positively perturbed, as in the cell adapts, we can get diseases like cancer. If the process is negatively perturbed, as in overwhelmed, we can get diseases like ALS. So blocking the UPR could overstress cancer cells and kill them, whilst activating the UPR could help neurons clear misfolded proteins. But how would we go about targeting such a mechanism? So if you remember, at the beginning we said that certain types of proteins, certain enzymes, have little pockets that include that are supposed to be fitting substrates, small molecules that bind to enzymes in order for them to carry out their function. And I'm sure you remember this little fellow. One of the transducers of the unfolded protein response is one such molecule. So in the little red box, you see an indentation next to the letter K. That's a pocket like that. And what we would be trying to do is make a drug that can fit the pocket instead of the substrate, but fit it much better in order to outcompete that substrate. And then either, depending on the nature of the molecule, either activate or block that enzyme. So this is one of the ways of finding a drug candidate. Because we would want something incredibly specific, because we wouldn't want to have any off-target effects, one way of having a template for a drug is to take a fragment of the target itself and then check if it fits in the pocket. Then, if it does, then we could convert it to a compound that is more stable, because protein fragments can be degraded, but mimics the fragment structure. So we are more certain that it can fit into that pocket. And then furthermore, we can find and map the binding characteristics of the compound on the enzyme, on that pocket, which means finding out what the bonds are, finding out the electrochemical and electrostatic interactions between these two, and thereafter, getting a good candidate out of those, we can use that map to search for more stable organic compounds in libraries of millions and millions of compounds. So, for example, the Zinc-15 database has 13 million compounds, and we can also search for already clinically approved molecules in order to figure out whether we could use already clinically approved drugs with a different clinical designation. So, in the end, we pick the candidate that is superior to the native substrate and test it in living systems. So this is a very quick video about how the new molecule would fit in a pocket. So this is the molecule that we spoke about before, the one with the little red box and the indentation next to the K. So that's what it actually looks like. The area in gray is that pocket, and the molecule in pink is the native substrate. So what we're trying to do is make a molecule, an organic molecule or a drug that fits that pocket better. So you can see that there are certain nooks and crannies, there are certain structural spaces that we can fit a little bit better with a molecule that we want to make. And we would want to do that in order for it to outcompete the native molecule, and we get something like this. You can see that it's a bit bigger, it fits better, and I don't know if you can see there are some black and yellow dotted lines. These are hydrogen bonds, so the more of those we have, the better, and more favorable electrostatic interactions. So once we've got that, we need to test it, to test this new drug, this new candidate on various types of models. So we can have cellular models, animal models, or even patient-derived models. So patient-derived models would be taking a biopsy, for example, and then keep it alive for a certain period of time in order to test drugs. And we'll be doing that to limit the use of animals and also have a testing mechanism that's a lot closer to the clinic rather than a model that is not human or a model that is just cells. So let's say that our drug has gone through all these processes and it works swimmingly, it works perfectly. So let's use them. Straight into the clinic? Absolutely not. We have to be very, very safe first. And as is the case with many things that sort of govern and inform good practice, they are informed by things that we've done wrong in the past and atrocities that have been carried out in the past. And clinical trials are no different. The clinical trial ethics and regulations are actually based on the Nuremberg trials and the doctor's trial in particular in 1946 to 1947, when medical practitioners and medical scientists were trialed for war crimes that included performing medical experiments without the subject's consent on prisoners of war and civilians of occupied countries, in the course of which experiments the defendants committed murders, brutalities, cruelties, tortures, atrocities, and other inhuman acts, as well as planning and performing the mass murder of prisoners of war and civilians of occupied countries, stigmatized as aged, insane, incurably ill, deformed, and so on, by gas, lethal injections, and diverse other means in nursing homes, hospitals, and asylums during the euthanasia program, and participating in the mass murder of concentration camp inmates. Now, those trials gave rise to something called the Nuremberg Codex, the Nuremberg Code, and that went through many, many iterations until 1964, when the World Medical Association came together to sign the Declaration of Helsinki, which was a statement of ethical principles to provide guidance to physicians and other participants in medical research involving human subjects. Now, that said, that is the duty of the physicians to promote and safeguard the health of the people. The physician's knowledge and conscience are dedicated to the fulfillment of this duty. And in medical research on human subjects, which clinical trials are, considerations related to the well-being of the human subject should take precedence of the interests of science and society. And despite how far we've come, vigilance, pharmacovigilance, is incredibly important, because at the end of the day, putting an experimental drug for the first time in people is very dangerous. And there are two very recent examples of that happening. One in 2004, which happened here in the UK at Northwick Park Hospital, where an immunomodulatory experimental drug was given to healthy volunteers and caused a cytokine storm. And a cytokine storm was a group of symptoms that was actually bandied about quite a lot during the COVID trial. And the 2004 to general trial left quite a mark. And this is something that people wanted to avoid, and because it caused really serious side effects such as gangrene. And you can see that on the poor lad's fingers at the bottom right corner of your screen. And in fact, BBC Two did quite a good documentary called The Drug Trial Emergency at the Hospital, which would be quite interesting for you to watch if you manage to get your hands on it. And then even more recently, in 2016, a Portuguese company produced a drug that was trialed by a contract research organization in France with a neural experimental drug, which also caused serious side effects, including one death. So that's why, although it takes less than a year, that second step, that clinical trial application, is exceptionally important, because, and this is why the application in clinical trial design looks like this. Now, I'm not going to go through the entire roadmap of clinical trial design and approval, because it's very complex, but this is the point. The take-home message from this is that it's incredibly difficult to actually get approval, and we have to meet certain criteria like adequate sponsorship, trial documentation, pharmacovigilance, and a very comprehensive ethics submission before we can even start a trial. And in the ethics submission, there are more and more iterations of them and more involvement from patients and the public in order to make sure that everything is centered around patient care. So these mechanisms keep evolving. And if you wanted to have any further sort of information about each and every single step, this roadmap is available on the MHRA website. And MHRA is the regulator for granting clinical trial licenses and approving medications in the UK. So if a trial is approved, then we'd go into trial designs. So there are a few different ones that can be used, but three common ones include the parallel, crossover, and 3+3. And there are subtle differences between them, but all of them are designed for different populations and are designed in order to give us unbiased approaches to, as well as safe approaches to clinical trial and drug development. So in the parallel design, we'd have a study population that would be randomized, and that would be done in a double-blind manner, which means that the patients themselves would not know whether they would be getting the placebo or the active drug, and at the same time, the physicians and the researchers that would be carrying out the trial would not know which drug or placebo the population has gotten. In this particular instance, we've got a number of people as 12, because that will probably be a phase 1 study. This is how few volunteers we get in phase 1 studies normally, between 6 and 12, and then they would be split in half randomly, and then one arm would get only the placebo in escalating doses, another arm would get only the active drug in escalated doses. In the crossover, you still get the randomization, but the placebo and active drug arm are interchangeable. So they will both get both placebo and the active drug with a period of washout in between in order to make sure that there aren't any residual effects from the previous dose. And then there are some trial designs that are actually more geared towards specific diseases. And this is why I've put cancer in brackets next to the 3+3 design. The reason for this is that certain diseases are quite a niche in a way, because in the case of cancer, it is widely accepted that cancer medication is very toxic, which means that a lot of the time the archetypal sort of design, like a parallel design, may not be needed when we're looking at a drug. So in these trials, the participants will all get the active drug, and they will get it in a manner of escalating dose. So the first 3 will get a particular dose of the drug, and then if that elicits no response or no positive effects, then another 3 will get a higher dose, and so on and so forth, until we get unfavorable toxicology or favorable results in order to figure out how the trial goes. So this is how one would go from testing a new drug, as we've described it, for example, targeting the unfolded protein response, all the way through to a clinical trial, and how one would go applying for a clinical trial and then going through it by consenting people in order to take part in the trial, and administering different doses of the drug in order to figure out whether it's effective or not. So usually these processes are carried out by many different centers around the world with different expertise. And I was very lucky throughout my career to be able to combine quite a few of these disciplines, having started my clinical work and clinical trial design and regulation in the UK, oncology research in France, computational biology and drug design in Sweden, biomarker discovery in Greece, and trying to merge all of these and put all this experience together in translational oncology back here at Oxford at the Nuffield Department of Surgical Sciences in prostate cancer in particular. And the reason why I'm giving you this whistle-stop tour of the past few years is that, as you remember at the very start of the presentation, we presented, especially the first part of the process, as something quite linear. So we've got a clinical problem, then we have some human data analysis, then maybe we find a biological target, and then we move into biological experimentation or animal humanized model experimentation, which, if it works, would lead into a clinical trial. But in reality, it actually looks a bit more like this. It's very circular. So all of these happen at the same time, and they all feed information into a circular manner in order to affect positively patient care. And one good example of that happening and of how it would positively impact clinical trial design and how how a medication or a vaccine would go through a clinical trial was the current situation when we saw a vaccine go through that incredibly laborious process of a clinical trial design and implementation in roughly a year, as opposed to what it normally would be, which would be about 15 to 16 years. So to summarize, proteins are essential for life. Protein production quality control can govern health and disease, and it can underlie certain diseases that could on the surface look completely unrelated. Targeting the unfolded protein response, which is what is trying to deal with defects in protein production, could be successful in a variety of diseases. But at the same time, pharmacovigilance is very important because the drug could very well work in an experimental model, but putting it into people is a completely different proposition. And the future of drug discovery may well be in drug repurposing and combination therapies, as a lot more information is being had from a lot of basic research. We get a lot more data coming in from different trials that show that different biological pathways are affected during therapy or even before therapy in the disease progression. And the more information we have, the more sort of treasures we can uncover of already clinically approved drugs that could be used for different diseases than the diseases that they have been attributed to. And this could lead to actually combination therapies of drugs that are not as harsh and can improve not only life expectancy, but quality of life. And this would be the crux of personalized medicine, a sort of tailor-made therapy for every single patient without blanket treating everyone. And the key to that are multidisciplinary collaborations. So that was my quick talk on how protein quality control governs health and disease. Thank you very much for having me again. And I'd like to thank the Atom Society and, of course, all the institutions that have supported this effort over the past few years, like the Nuffield Department of Surgical Sciences, Chemistry and Progenesis Stress Signaling in France, the University of Gothenburg, and INEOS Inspired Operational Systems in Athens, as well, of course, the NHS Hospitals Trust here in Oxford. Thank you very much, and I'd be glad to take some questions.
Thank you very much, Demetrius. That was a really interesting talk, a whistle-stop tour on many topics, but still managing to be quite detailed and very interesting indeed. Yes, if anyone has any questions, please pop them in the chat on the YouTube channel, and I'll be able to bring them up. While we wait, I can ask a couple questions. So you mentioned very briefly the nature of clinical trials, actually sort of all the different areas feed into each other throughout the trial rather than it being linear. But given that it does, even though it's normally like that, it does normally still take well over a decade. The elephant in the room and the question that a lot of people have been asking in the last few months is, how is it that the trial this year did manage to only take a year? Can you maybe, as someone with a lot more experience, touch on that?
Yeah, absolutely. I mean, the fact that we have reached a vaccine so quickly is firstly because we were lucky enough to have a lot of that first part that's let's say let me get onto that slide of this already in place. So a lot of that had already been researched. So those 3 to 6 years were out of the way already. The clinical trial application was fast-tracked. So it meant that we were able to go through the regulatory processes very quickly. So the red tape was actually slashed completely. And then the nuance of clinical trials actually depends on the question asked as well. So in a very accelerated trial design, really traditional primary objectives like severe hospitalization or like death are not taken into consideration. So it's not, so in these particular trials, we were trying to see whether there was an immune response rather than trying to figure out whether the vaccines could prevent death. That type of trial, which is a lot of the time what trials we see in cancer, for example, would take years because the follow-up is huge. And also in a pandemic, it's very difficult to carry out a trial like that because you don't want to put people more at risk than they already are when they come into the clinic to actually give blood or any information at all. So there are certain nuances, and also the fact that we would be able to count on a lot of basic research, drug development, and preclinical translational research work already happening, which kind of highlights the value of it because it was ready. If it wasn't, this whole pandemic would have been a completely different proposition.
Thank you. We have a question from Jane who asks, is the UPR taking over in all cells? And if so, will targeting IRE affect the ability of healthy cells to mount a response?
Very, very good question. So you sort of hit the nail on the head there. So yes, the UPR is taking over in all cells. And the again, we're dealing with a very finely balanced nuanced system because what the UPR is a homeostatic mechanism. Homeostasis is the biological process of making sure the body oscillates around a given balance. So it can't go too far one way or too far the other. So mechanisms like that make it healthy most of the time. So targeting the UPR is incredibly difficult because, as you say, it could affect all the cells in the body. Now, what could overcome this is that the cells that we are targeting, and as such, the doses of the drugs that we are testing, are not like normal cells. Cancer cells are not like normal cells. So maybe what we would be trying to do is that alongside chemotherapy and radiotherapy, we would be giving a dose of that of those inhibitors, let's say, in order to sensitize cancer to the already existing therapies. So we make it more receptive to the existing therapy rather than giving a massive dose of something like that and then affecting the whole body. So it is a very difficult mechanism to target, but because it's so fundamental to life, it would have a much bigger positive effect if we manage to do it properly. And the basic research behind it, which gives us a more stratified approach to patients, so we cannot give that to everyone. That would be probably irresponsible because not everyone will respond the same. Not everyone has the same UPR activation profile. We would be able to conduct clinical trials in a very targeted manner.
It's really moving away from the one-size-fits-all approach.
Oh, yes. Yeah.
Thank you very much. We've got a question from Andy who asks, will the newly discovered ability to form accurate 3D computer models from the protein's primary sequence transform your research? I think this is referring to something that was announced a few months, tail end of last year.
Yeah, definitely. Yeah, absolutely. Because currently, we can only do that on crystallographic models that have already been generated. Now, to generate the crystal structure of a protein is no mean feat. It requires a lot of specialized equipment, and the way that it's done is fiendishly clever, and that's why the Nobel Prize was given to it in 2017, I think. But that also means that we're kind of limited in terms of the targets that we can affect, because if someone has made the crystal structure, then we can do the work that I showed during the presentation. If they haven't, we're kind of blind. So predicting 3D computer models would be really useful. And of course, not in isolation, because the one thing that I didn't say during the presentation, but is true, is that these proteins are actually flexible while those models are rigid. So they form a guide for us to design drugs, but then the next step, testing them in living cells, in living organisms, is paramount, is very, very important, because then we'll find out whether it actually works or not.
Yeah, thank you very much. I've had a question come in from the committee. I don't have the functionality to show it on the screen, I'm afraid, but I can read it out. So this has come from Susan, who asks, what makes the genetic blueprint go wrong in the first place? Would you describe the process of regulating protein production as a form of homeostasis?
Yeah, brilliant question. I mean, I think I already said in a previous answer that the process of regulating protein production is homeostasis. It is a homeostatic mechanism. And the genetic blueprint is the problems that arise with it can be attributed to many things, from chance to a particular event throughout the cell's life, or exposure to environmental factors. So, for example, radiation itself is mutagen, so it breaks DNA. So when DNA breaks, then there is another mechanism called the DNA damage response, which tries to actually glue it back together as best as possible. Now, that doesn't always work as well as it should. If you glue it back not in the correct way, then the code, the blueprint, is wrong, and therefore you start making wrong proteins. Sometimes there will have been a founder effect at some point. A mutation will have happened in a particular gene, and then it will be passed on through generations, and that's how you get diseases that are attributed to single mutations, like Huntington's, for example, or like cystic fibrosis, where you will have some mutations that give a faulty protein. So in cystic fibrosis, it's a chloride transporter that cannot pump chloride through your lungs properly, and that means that mucus cannot travel from the bottom of your lungs to the top in order to expel it, and that's why you get respiratory problems. In Huntington's, obviously, you get those horrible chorea movements and so on. And then the other reason why you would be getting DNA breaking and therefore more chance of mutation is in cases where you have an increased production of cells. And at one point in an increased production of cells, which could be immune cells or it could be blood cells, for example, something will break and something will not be made correctly, will not be remade correctly, and that can give rise to cancer. So leukemia, for example, is a blood cancer where there is an accumulation of faulty cells and so on and so forth.
Thank you very much. It does not look like we have any more questions coming through on the chat. So I guess with that, I'd like to thank you again, Doultsinos, for a really interesting talk and some really nice answers to the questions as well.
Thanks
to
for having
everyone else from Atom, next month we have Dr. Ryan Ershad coming to talk to us about Mars and why it continues to hold our fascination, which will coincide nicely with the first of 3 Mars landings that are due to happen this year. Hopefully you'll all be able to join in, and we'll see you then. Bye.
Thanks. Bye.